Hello, friends. If you remember, last day we were discussing about some MCQ based theory part on the chapter Biotechnology and Genetic Engineering. So, we'll revise once again. So, what we were studying? We were studying about the palindromic DNA sequences. Okay, the palindromic DNA sequences, what are they? Palindromic DNA sequences are which can be read same from the left and right. So, if you read from 5 prime to 3 prime, it is GAATTC, and when you read from 3 prime to 5 prime, okay, it could be read in the same way. Okay, they are kind of same sequence when it is read forward, that is left to right, or backward, that is from left to backward, from right to left, from a central axis of symmetry. So this, you can say, this is the axis of symmetry. Okay, now we also read about restriction endonuclease. Yes, do you remember what is restriction endonuclease? So these are the enzymes, say for example EcoR1, and where is this EcoR1 found? This EcoR1 enzyme is found in it is a colon bacteria. It is a colon bacteria, and here EcoR1, if we understand the naming of this restriction enzyme EcoR1, this is a colon bacteria, and this enzyme is found in E. coli. Okay, this enzyme is found in the colon bacteria E. coli. This colon bacteria E. coli is found in the colon, and here CO, here CO stands for what? Here CO stands for coli. Okay, CO stands for coli, E stands for Escherichia, and CO stands for coli. And the first letter used for the enzyme is the first letter of the genus, and the second and the third letter here it is for the species. Okay, and the first letter here it is written in like it will be written in italics. Okay, and next is the strain of the organism. So R1 is the strain of the organism. So at last is a Roman numerical. This is a Roman numerical which signify the order of discovery. So this is the order of discovery. Order of discovery. Okay, this is the order of discovery. So the enzyme EcoR1 was isolated from the bacteria Escherichia coli, which is CO stands for coli, and strain is RY13. Where this R comes from? It comes from strain RY13. Okay, which is for R. And it was the first endonuclease, which is one isolated from E. coli. Okay, so this is the revision of restriction endonuclease. Next, we have like known something about transgenic crops. Okay, we have studied a very little about transgenic organisms. So transgenic organisms are what? Here, the transgenic organisms are what? They are kind of animals. So transgenic plants we have discussed about tomato. When we discuss about transgenic animals, so let me write about transgenic animals now. Transgenic animals. Okay, so in case of transgenic animals, these are the animals that have their DNA manipulated. The DNA is manipulated so that extra foreign gene would be there. Okay, this is the DNA. If I consider this is a plasmid, this is a foreign DNA. If this is a linear DNA, animals have generally linear DNA. So this is the foreign gene. So this foreign gene which have in the organism is called transgenic organism, and these transgenic animals like mice, rabbits, pigs, sheep, cows, and fish, they have been already produced, and the 95% of all existing transgenic animals, they are mice. So, okay, most common are mice. Okay, so this was our 15 number. So next, again, we studied about like whenever any transformation occurs, it is checked on the gel whether whatever we have done is like foreign gene have been inserted or not, has been multiplied or not. So every step in the cloning process, it is confirmed by gel electrophoresis. Okay, to check the size of the DNA fragment, presence of the DNA fragment. Okay, so the gel electrophoresis here, what happens in the gel electrophoresis? The DNA fragments which are formed, if this is a gel, so these are the wells. Okay, so the fragments are resolved. So whether the fragment, target fragment is present or not, this can be checked by gel electrophoresis, and so fragments are separated on the basis of the DNA fragments, they are separated on the basis of size. Okay, so what is the polymer used? Here agarose is used. Okay, and agarose extracted from seaweeds. Extracted from seaweeds. Okay, and this is used as a polymer in agarose gel electrophoresis. Okay, next we come to kind of another very important topic, that RNA interference. So what is RNA interference? Known as RNAi, it's a technique. Okay, what happens in this technique is, so I'll explain you this with an example. So nematode, several nematode parasites, okay, they parasite on a wide range of plant and animals, also human beings. Okay, so example, Meloidogyne incognita. What it does, it infects the roots of tobacco plants. It infects the roots of tobacco plants and causes reduction in yield of the tobacco. So there, this RNA interference is a novel strategy which was adopted to prevent this infestation, which was based on the process of this RNAi interference. And this RNAi interference, it silences some of the genes in this Meloidogyne incognita so that they cannot infect the tobacco plant. Okay, and this RNA interference, this is common in eukaryotes. Takes place in eukaryotes or eukaryotic organisms, and it is a method of defense. Okay, so we will elaborate more on this later on. Okay, so if I revise once again about the Bacillus thuringiensis, if you remember. I hope you remember. So, this Bacillus thuringiensis is what? Bacillus thuringiensis is a bacteria. Okay, again, we are revising. This Bacillus thuringiensis, it's a soil-borne bacteria. It's a soil-borne bacteria. Okay, and it produces some proteins. It produces some proteins. What proteins? Bt toxin. Okay, it produces some proteins that kill certain insects. What group of insects? Lepidopteran, coleopteran, and dipteran. Okay, lepidopterans are what? Lepidopterans are, they are budworm, armyworm, budworm, armyworm. Coleopterans are what? Coleopterans are beetles. Okay, coleopterans are what? They are beetles. And dipterans are what? Dipterans are what? Dipterans are, they are flies and mosquitoes. Okay, so Lepidopteran is budworm, armyworm, coleopteran is beetle, and dipterans are flies or mosquitoes. Okay, so these toxins, okay, these crystals, they contain a toxin. So this forms some kind of Bt toxin, which are kind of crystals. Okay, so these crystals, they contain a toxic insecticidal protein. Okay, this toxin does not kill the bacterium because this bacterium has an inactive protoxin in them. Okay, this Bt toxin is released in the form of protoxin. Okay, this is released in the form of protoxin so that it does not harm the bacterium. But this protoxin, when it is ingested into the pest or the insect, when the insect ingests this protoxin gets converted to the active form. Okay, protoxin is converted to the active form. Active toxin. Okay, protoxin is converted into active toxin. As a result, what will happen and how this active toxin is formed? So once an insect ingests, it is converted into the active form, and due to the alkaline pH of the alimentary canal. So inside the gut of the insect, the pH is alkaline. So due to alkaline pH, protoxin is converted into active toxin in the alimentary canal of the insect, and this activated toxin, it binds to the surface of the midgut. Okay, so I'm explaining it here only because it would be easier for you. So I'm elaborating it here. Okay, so in case of Bacillus thuringiensis, it releases protoxin. This protoxin is when ingested by insect. In that case, protoxin is converted to active toxin because of the alkaline pH in alimentary canal of insect. Once active toxin is formed, what happens? This active toxin, this binds to the surface of the midgut epithelial cells. Okay, and then create pores. Okay, and these pores cause swelling and lysis and death of insect. Understood the whole process? Okay, the spore causes swelling and lysis and death of the insect. Clear? So we were discussing about a nematode, okay, which through RNAi mediated mechanism can uh, not allow or can inhibit the parasitic activity. So basically, this nematodes parasite wide variety of plants and animals and human beings, and this infects the root of the tobacco plants, and it causes the reduction in yield we have already discussed. And there is a novel strategy which was adopted, and this method involves what? Silencing of. So what this method do? This method involves silencing of specific mRNA. Okay, specific mRNA due to complementary double-stranded RNA molecule that binds to and prevents translation of mRNA. So what happened? This RNA interference, some kind of double-stranded RNA, what happens? Some kind of uh, complementary, this when this RNA is introduced, this is complementary to the nematode RNA, and when this RNA I, when it binds to the nematode RNA, in that case what it is going to do, it will silence this particular RNA. Okay, so how this will happen? The Using agrobacterium vectors, nematode specific genes were introduced into the host plant. Okay, so inside the agrobacterium, nematode specific gene, nematode specific gene, which is complementary to the nematode, it is introduced into the host plant through agrobacterium mediated. Okay, when it is introduced into the host plant, as a result of which what will happen, that it produces both sense and antisense RNA. It produces both sense and antisense RNA in the host cells. Okay, and these two RNA, which is sense and antisense RNA, they being complementary to each other, it forms double-stranded RNA that initiated RNAi, and thus silence specific mRNA of nematode. Okay, so what happens? The introduction, so when this is introduced into the host plant, this inside the host plant, it can produce both sense and antisense RNA. So either this one or this one, they separate and they bind to the RNA of the nematode, and when it binds to the RNA of the nematode, it does not allow the RNA of nematode to express, as a result of which the mRNA of nematode will be silenced. Okay, so this consequence was that the parasite could not survive, and inside the host plant the parasite could not survive, and so this agrobacterium mediated, when this new gene is introduced into the host plant, the host plant became transgenic. So these transgenic plant having nematode specific gene, it silences some of the nematode mRNA, by binding to it, by becoming, because this gene is complementary. This gene is complementary to nematode gene. So it goes and binds to the nematode gene and silence it, as a result of which the nematode cannot survive in the transgenic host expressing this specific interfering RNA. So transgenic plant therefore gets protected. Okay, nematode separated. Nematode cannot survive. Okay, so this is how this nematode can be protected. Got it? So another question comes like, another question is, first transgenic cow Rosie. So first transgenic cow was what? First transgenic cow was introduced in 1997. Okay, was introduced in 1997. Its name was Rosie. Okay, what it produced? It produced human protein enriched milk. Okay, and this contained what? This contained alpha lactalbumin, alpha lactalbumin, and this was nutritionally more balanced product for human babies than for natural cow milk. And isolation of the DNA from other macromolecule, it is achieved by treating the bacterial cells or animal tissue with enzymes. So with lysozyme. So basically what? The first transgenic cow, the milk contained the human's lactalbumin. This is very important, you need to know. And this is more nutritious for the human babies. Okay. So this cow produced some proteins which is very rich in proteins required by the humans. Okay. Coming to the next part. So, microbes have been used for commercial production of certain chemicals like organic acid and alcohol and enzymes. So, there are certain kind of microbes which are used for producing some commercial products. Say, for example, Aspergillus niger. So, what is Aspergillus niger? It is Aspergillus niger is what? Aspergillus niger is a kind of fungus. Okay, it's an acid producing fungus. Okay, next is what kind of acid? Citric acid. Next is Acetobacter aceti. As the name says, bacter is bacteria and produces aceti is acetic acid. Next is Clostridium butylicum. Clostridium is a bacteria, produces what? Butyric acid. Next is Lactobacillus. Bacillus is what? Bacillus is bacterium, produces lactic acid. Okay, interesting to know that, right? So, another important one is human insulin, which we have already discussed earlier. So, human insulin, it is made up of 51 amino acids. Okay, human insulin, it is made up of 51 amino acids. Clear? And this is arranged in two polypeptide chains, chain A and chain B. A chain having 21 amino acids Okay, and B chain having 30 amino acids. B chain is longer. And the hormone, the insulin hormone develops from a storage product called proinsulin. So I write it here, proinsulin forms insulin. Okay, proinsulin has three chains, A, B and C chain. And the C chain has 33 amino acid. This has 21, this has 30 and this has 33 amino acids, which is removed. So C peptide is removed. Okay, I have told you in the last video also that pro-hormone or pro-insulin has extra stretch of C peptide. So 33 amino acid is removed for insulin formation and the C peptide is not present in the mature insulin. It is removed during maturation into insulin. Clear? So again, moving forward. So we were discussing about DNA fingerprinting. Okay, the satellite DNA was used in forensic science. We were studying about DNA fingerprinting, which is a technique for identifying repeated sequences. So DNA fingerprinting is what? It is a technique for identifying repetition in the sequences, identifying repeated sequences. Okay, in human genome, okay, and it produces a pattern of bands unique for every individual. Okay, it shows some pattern of bands which are unique to everyone, which are same in a particular generation, which is unique to every individual. Okay, so it was first used by Sir William Herschel. Sir William Herschel as a method of identification. Okay, and these fingerprintings they prepared from minute amount of blood, semen or hair bulb. Okay, and this DNA is extracted from the cell. Okay, in high speed refrigerated centrifuge there is a proper method for DNA isolation. And if DNA content is limited, it can be amplified in the PCR machine by polymerase chain reaction. And for this, Taq polymerase is used, which we also discussed last day. This Taq polymerase have the ability to survive temperature greater than 90 degrees centigrade, and it is can be isolated from the bacteria, basically archaea bacteria, uh, Thermus aquaticus. Okay, so this was all about for today's class. We will continue the revision part and the new topic in the next video. If this video is helping you to revise and gain knowledge, because what I will be doing, I will be covering each and every topic that is important from this chapter. And while doing that, I will keep revising the old topics and I will make you uh, well prepared in each of the chapters by successive videos. Just you have to do like and share the video. And if you have any questions, you can always comment in the comment box. Thank you so much.